Hello, everyone. People are still joining, so we'll give it a minute or so before I hand over to today's speaker. As we get ready to start, maybe you can share in the chat where you are tuning in from today. It's always interesting to see how far we reach and who we reach with these seminars. While you do that, maybe a bit of housekeeping. Please be reminded that the seminar is being recorded and that the recording will be available on the SCB Vimeo page within a day or so. And you can, of course, also access all of our previous seminars in the Emerging Issues in Conservation Seminar series on the SCB's Vimeo page. I would like to request that you please keep your microphones muted and your cameras off for the duration of the seminar. At the end of the seminar, you're welcome to unmute when we have a question and answer session. And at that stage, you are welcome to raise your hand and ask questions, maybe get some discussion going if you can. During the seminar, you're also welcome to post any comments and or questions in the chat. So my name is Martin Strauss, and I'm hosting the seminar today. I'm a senior lecturer in the Department of Environmental Sciences at the University of South Africa, and I'm also an associate editor at Conservation Science and Practice, and it is in that capacity that I've been organizing the seminars for 2021. So welcome to what is now our 26th seminar of 2021. In the series, we feature articles selected by the editors that are recently accepted in early online view or recently published in the 3 SCB journals, those being Conservation Biology, Conservation Letters, and of course, Conservation Science and Practice. Please visit the SCB Emerging Issues in Conservation Seminar Series webpage for a list of upcoming seminars through September. We're busy identifying potential seminars for October through December. So if there's perhaps a recent or early view paper in one of the 3 SCB journals that you would like to suggest we feature, you are welcome to let me know, and I will run it past the editors. As soon as our speaker starts, I'll paste my email address in the chat, and you can send me a suggested paper if you would like. So it is my great pleasure to then introduce you to our speaker today, Dr. Theresa Laverty. Theresa received her BA in Ecology and Evolutionary Biology from Princeton in 2010. Her PhD in Fish, Wildlife and Conservation Biology from Colorado State University in 2019. For the last 2 years, she was a postdoc fellow at Colorado State, where she studied the population ecology of migratory long-nosed bats in relation to the timing and availability of their nectar food in New Mexico. As of this morning, Theresa is a new postdoc scholar in the Brook Lab in the Department of Ecology and Evolution at the University of Chicago, where her research will focus on the population and community ecology of Malagasy fruit bats. In this new position, Theresa looks forward to expanding her research applications to include those relevant to the knowledge and management of wildlife and zoonotic diseases. While her fieldwork over the last 8 years is largely focused on bats, her findings are broadly applicable to a range of taxa. And in addition to her ecological research, she's also led the design and analysis of semi-structured interviews to gain a broader understanding of the conservation challenges and opportunities in her study areas, and advocates for diversity, equity, and inclusion within STEM fields. Today, Theresa will talk about her paper, Early View in Conservation and Biology, entitled Indirect Effects of African Megaherbivore Conservation on Bat Diversity in the World's Oldest Desert. And with that, Theresa, over to you.
Hi, everyone. Thank you, Martin. It's very nice to join you all and to be talking today about the paper that's in Early View at Conservation Biology, entitled Indirect Effects of African Megaherbivore Conservation on Bat Diversity in the World's Oldest Desert. And this is co-authored with Joel Berger. So as a community ecologist, I've been particularly interested in basically where we find species and where we don't find species and why they exist where they are in the world today. And typically what I think of when I hear community ecology is our traditional food web systems, where you have predators at the top of these food chains that are feeding on primary and secondary consumers, and plant productivity and your detrivores down at the bottom of these food webs. And one of the most famous food web systems, when we think about this, is Robert Payne's work out of the intertidal zones of Washington here in the United States, where you have piaster, or these sea stars, that were feeding on many levels of the food web in intertidal zones. And when these sea stars were removed, bivalves, or particularly these mussels, were taking over parts of the food web. And so Bob Payne coined sea stars as keystone species in 1969. But to be honest, the work that really got me interested in this project was some of Bob Payne's much more recent work that recognized the rampant indirect effects of mammalian herbivores rather than predators in food webs. And in this paper from 2000, he states that grazing mammals, because of their size and often conspicuous abundance, are likely to play significant roles in community organization. And this was particularly interesting to me because many of the world's largest herbivores are in decline. And so I was really curious how these species may be functioning in systems that still have these herbivore communities quite intact. And when we think of large herbivores, we don't typically think of desert ecosystems, particularly because deserts are rarely recognized to be very abiotically limited. As Darwin stated in his Origin of Species, when we reach absolute deserts, the struggle for life is almost exclusively with the elements. And it's not until we reach the extreme confines of life in Arctic regions or the borders of an utter desert will competition cease. And so these desert ecosystems are not typically thought of as biodiversity hotspots. They're thought to be particularly limited by the amount of rainfall that they receive. But nowhere is competition for resources as intense as in desert ecosystems. Deserts and rangelands cover about 1/3 of the Earth's terrestrial surface area, and they're highly variable in the amount of annual rainfall that they receive. As you might notice, this map of the coefficient of variation of interannual precipitation closely overlays the global distribution of deserts from before. So in these extreme ecosystems, we find these boom and bust economies and highly ephemeral resources, where abiotic and biotic factors can be independent of one another or interact in how they shape the distribution of desert life. And for today's talk, I'm going to be focused on the Namib Desert, which runs from southern Angola to northern South Africa on the Atlantic coast. My particular study area was in the northwestern corner of Namibia in the Kunene region, just to the east of Skeleton Coast National Park. The Namib Desert is considered to be one of the world's oldest deserts. And what was really great about working in a country like Namibia, a very recent country, is that few countries put terms like conservation or biological diversity in their constitution, but like Bhutan, Namibia does. So I was particularly interested in how this old desert may have shaped how species interact in these ecosystems. And when most people think of the Namib, they think of a scene like this, where you have some of the world's largest sand dunes standing behind dead trees that died about 1,000 years ago. This is a scene from Sossusvlei in the southern Namib. However, the northern Namib looks something more like this, where you have these really rocky, mountainous regions that are interspersed by ephemeral river systems. And it's in these areas where you'll see quite a bit of vegetation. So here's another scene of the Namib. But as I mentioned before, I was really interested in working in a system with an intact herbivore community. The Namib, the northern Namib in particular, is home to not only the African elephant and the Angolan giraffe, but also the largest free-ranging population of the black rhinoceros, in addition to many smaller herbivores and pastoralists and their livestock in this ecosystem. So the Namib is really characterized by these ephemeral river systems, which for the most part look like this. However, you're lucky if you're there for a handful to perhaps 20 days a year. You may see some of these rivers sustain above ground flows. And this is due to precipitation that's oftentimes received at their headwaters, many kilometers away from where these rivers are actually lying in the desert. And these act to really strongly impact the seasonal differences that we can see in these desert systems. So not only does localized rainfall, but also rainfall from much further away in the more savanna-like communities affect the patterns of diversity and distributions of species in the Namib. Oops, sorry. However, the more permanent forms of water in this system exist as natural springs on the left-hand side of your screen, or as artificial pools on the right-hand side of your screen. And these artificial pools or boreholes in the system were actually constructed to try to keep large mammals like elephants away from human settlements in these pastoralist communities. But no matter the type of water body, these really act to concentrate not just a large number of species in this ecosystem, but a large number of individuals, particularly during dry seasons or drought years. And this isn't just true for diurnal animals as pictured here in Etosha National Park, but also for our nocturnal mammals like bats. So even though many desert adapted bats are less restricted in the amount of water that they need to receive each day, one of the first things that many bats will do as they wake up at night is to visit a water hole and grab a couple of drinks. Bats all drink on the fly, and they then will sometimes stay at these water holes to forage for insects, or they may travel elsewhere. But these water holes really do act to concentrate bat biodiversity. And as a community ecologist, working with bats is our really cool study system in that there's over 1,400 species of bats recognized in the world. And so that means about 1 in 5 mammals are bats. So even in areas like desert ecosystems where we don't traditionally assume there's a large number of species, you still have quite a number of bats that are interacting across this landscape. So that kind of led to my question as to what roles do large herbivores play in structuring desert bat communities? And for this study system in the Namib, I was particularly interested in how herbivores, by clustering around bodies of water, may be affecting bat communities. And before I get too far, I just want to highlight some of the key team players for this project, which included my colleagues at the University of Namibia, as well as Sesfontein Conservancy, and my co-author Joel Berger, as well as a field assistant Mallory Davies here from the United States. So when we think of these desert ecosystems, I've mentioned that they're generally abiotically limited. And so it's generally thought that many species aren't reaching high densities in the system that would have impacts on the vegetation community around them. However, herbivores in deserts have larger effects on the vegetation closer to water than further from water. And this is known as a biosphere effect. And a study done earlier in the NAMIB by Keith Leggett and others in 2003 found that grazing pressure near water sources has altered the density, species composition, and biomass of vegetation in about a 2-kilometer sacrifice zone. So it's that area that's immediately adjacent to water bodies that's really being affected. However, beyond that high impact zone, vegetation's more dependent on the annual rainfall than on land use, and thus behaves more in a non-equilibrial manner as predicted in desert ecosystems. So for this study, I was particularly interested in how herbivores, by clustering around water bodies, are affecting vegetation. And this in turn can affect the insect community in these ecosystems, particularly for aquatic insects, but also your terrestrial insects that are reliant on many of these vegetation plant communities around water for habitat or for food. And then this in turn can of course affect the insectivore level of the food web. And for this particular study, I was interested in studying bats for the reasons I listed earlier, as well as we know quite a bit more about birds and lizards in these ecosystems, but many African bats are still understudied. So I thought this would be a great way to be able to contribute to both the natural history of African bats and understand how this community was interacting. So for my research, I was particularly interested in how megaherbivores may be affecting bats either directly or indirectly via their effects on vegetation. And this is all centered around bodies of water. Now, megaherbivores, rather than just large herbivores, are particularly interesting because on a per individual basis, these species eat way more vegetation than other species. And elephants in particular are known to be able to really shape the 3D structure of plants. So here you're able to see elephants and giraffes establish a very strong browse line on ana trees in the Waneb River in the northern Namib. However, they're also able to really shape the vegetation at much shorter levels, like smaller mopani trees, by debarking, which can essentially kill a tree if it's done well enough, or by pulling branches and basically reshaping these bushes. They're kind of our landscape architects in this area. And as I mentioned, there's also the Angolan giraffes present in the northern Namib, as well as the largest free-ranging population of the black rhinoceros. These tend to, the rhinos tend to exist in the smaller dry river channels. However, really these 3 species are all interacting at these same water holes. So I was particularly interested in how does large herbivore or megaherbivore activity around these water holes vary seasonally, and how could this affect bats? To look at megaherbivore activity, I was conducting dung transects, where I established 3 120-meter long by 4-meter wide belt transects around different sites. And then I revisited these sites each month to identify, count, and remove all dung piles to get some sort of indicator of megaherbivore activity or use of a site from month to month. And while I sampled 23 bodies of water, only 16 of these sites were actually surveyed for both bats and megaherbivore activity. And this was largely due to restrictions for my ability to reach the next site within a night, but also occasionally due to the fact that a few of these sites were either at a conservancy office in town, or one or two of these was also at a tourism lodge. And so I had some restrictions to my sampling based around that. So for just megaherbivore activity, what you're seeing here is the mean site level megaherbivore use on the y-axis, estimated from monthly dung surveys in northwestern Namibia over the dry season in light gray. And this was data I collected between mid-November 2016 and late January 2017. And then also the wet season in dark gray, which were from data collected from early February through early April 2017. And what you can see here is that most of the herbivore activity in this landscape was dominated by elephant and giraffe at these water holes. There are some black rhinos that are detected at these sites. However, a lot of times rhinos are more reliant on these dung midden piles. And so where you may find feces is much more related to a midden pile for them than for elephants and giraffes. However, I was able to also look at some of the camera trap photos taken by other researchers around these water bodies and confirm that, in fact, there is a lot more elephant and giraffe use at these sites. I then was interested in how elephants were affecting the vegetation productivity at these areas. And ideally, I would have been able to go out there and do a huge project where I was looking at the vegetation cover and density across these different sites with things like cover boards and densiometers. But due to restrictions of being on foot among some of these species and simply time, I instead was relying more on satellite imagery for this piece, where we were calculating the normalized difference vegetation index for the dry and the wet seasons based on those same dates as before. And we calculated the mean NDVI within a 2-kilometer buffer around each body of water. We did do some model selection to see which buffer size made the most sense here. And in fact, the 2-kilometer matched up quite well with Keith Leggett's work from before. So that's what we ended up sticking with. And it was also the buffer size that was showing the greatest impacts on bats. And then for the dry season, you'll notice what I'm going to be presenting here is the NDVI values in darker and lighter greens, where darker greens indicates a higher productivity. And if you focus on the words around Sesfontein and Pollenbog, I don't know if you can see my pointer, but in the top center of your screen, you'll notice that between the dry and the wet seasons, there's quite a bit of difference. And mean NDVI around these bodies of water was approximately 60% greater in the wet season than in the dry season. And that's in those buffers around each of those points you'll see on the screen. You'll notice that all of those were bodies of water. And here, the black color represents artificial pools, while white represents natural springs. So I had both across this landscape. And then lastly, I was sampling the bat community that uses these bodies of water. And I was doing that through acoustic sampling. Acoustic surveys are really great because you're able to determine based on the call, shape, size, and duration, oftentimes which species are present and how active they are at these sites. So in this example here, you're seeing on the right-hand side is from a North American system where the big brown bat was being recorded. And you can see each individual call as a bat passes the microphone. In the NAMIB, here's a couple of examples of different species we encountered and their calls. So you can really see that the frequency and duration and actually the shape of the call can really vary from species to species or from family to family. And while we do not have an acoustic call library available for the bats of Namibia, this is something that I would love to work on throughout my career and hope to kind of contribute to, at least for this system. I ended up doing a cluster analysis and then individually went through each of these calls and assigned them to species. We were able to determine that there were 16 different species present in the system. And here's just an example of their spectrograms here. And from these data, we're able to look at the amount of bat activity on the y-axis. And this is activity and not number of bats, because we're never sure if it's one bat that's flying back and forth in front of a microphone calling, or whether it's a whole group of bats that's flying by just once. And then we look at activity over time. And so in that example on the left-hand side of your screen, you'll see peaks for 4 different species, 2 that were very active just after sunset and 2 that were active at a lesser extent at different times in the night. And bat activity is usually used in our research to kind of indicate which sites are most important for bats. And this could be because they were close to a roost, or it could be because it's a great foraging spot. And as we said, we're not really sure if this relates directly to the number of bats that use a site, but generally, if there's higher activity, these sites tend to have higher conservation value. So I was recording bats for an entire night at these 23 sites of sampling. And we did this during the dry season and during the wet season. So for each study site, I had 1 full night of sampling for each season. Let's see. Now, just to double check that we're identifying bats accurately, this study was conducted during a 1.5 years of research in the Namib. And so I was also looking at bats through direct capture and mist nets to confirm species identities and record hand release calls. But there were many benefits to using acoustic sampling for an all-night survey. In our study system, it was not uncommon to come across species like elephants or even lions at the mist net. And sometimes they would tamper with more long-term equipment that we had out there. Rhinos would also occasionally approach the net if they were unaware that we were present at the time. And so any way that we could minimize the disturbance to the wildlife communities that really rely on these bodies of water was a great way to do any further sampling. But to give you an idea of what some of these species look like, here's just a handful of the insectivorous bats that we captured. And I'd like to highlight that there are a couple of fruit bats that can be found in the Namib Desert. These are usually quite rare. And for Eidolon, they really only come in during the winter months when the east winds hit the area. However, these species do not echolocate, and we were really interested in insectivorous bats for this study. So while we did capture some of these during other times of the year, they're not included in our analysis here. So over the 46 nights of sampling, it was 23 sites recorded once in the dry season and once in the wet season. We recorded about 120,000 bat passes, and that included 16 different species. And when we look at the species richness at a site during the dry and wet seasons on the x-axis, For artificial pools in blue and natural springs in orange, you'll notice that species richness didn't really differ among seasons or these types of water bodies. However, when we look now at mean activity, or the number of bat passes at a site for, again, the dry and wet seasons, you'll notice that artificial pools harbor about twice as much bat activity as natural springs, regardless of season, and that the wet season harbored about twice as much activity as the dry season. However, through a study we did with previously published in Biological Conservation, we found that these differences were not due to differences in water quality per se. And so we were quite interested in what are the other factors that could be shaping the bat community in this area, including those biotic interactions with elephants or giraffe or rhinos, these megaherbivores. So to look at these relationships, we use structural equation modeling. It's a form of path analysis that allows you to look at how different variables react simultaneously rather than using different univariate relationships. And specifically, we used the package Lavon in the program R to do our analysis, but we basically designed 3 different models where the relationships between bats and megaherbivores were either direct, as you see in the top left of your screen, or mediated entirely through vegetation productivity, as you see in the top right of your screen, or they had both direct in orange and indirect effects in blue present between these megaherbivores and bats. So we were interested in how megaherbivores were affecting bat communities and looking specifically at bat overall activity and species richness. We then used maximum likelihood framework to select the top model for each season using AIC model comparison. So in other words, for each season, the dry and wet season, we took data associated with each of these factors, ran structural equation models, and then we're selecting the top model with AIC model comparison. For the dry season, the indirect effects model was the top model. And what you're seeing here are the partial regression coefficients with R² values representing the amount of variation explained for each variable up until that point in the model. The black arrows are representing statistically significant partial regression coefficients, and gray, as you'll see in just a bit here, will represent statistically insignificant relationships. So for bat species richness and activity, the most supported pathway was an indirect relationship with megaherbivores, where megaherbivores and vegetation productivity had a positive relationship. This could either be due to megaherbivores trimming vegetation in a way that stimulated growth, or due to megaherbivores actually clustering in areas where there's greener vegetation. And then vegetation productivity, in turn, had a positive relationship with bat species richness, which is what was predicted with other studies. Basically, other studies have shown that insect abundance tends to increase with NDVI. And so this relationship is probably due to the fact that bats are more diverse in areas with greater food availability. However, it may also be reflecting the differences that megaherbivores create in the 3D structure of plant communities, allowing less maneuverable bats to access sites. So one thing worth noting here is when we look at overall bat activity, it's really kind of a black box in the system. And bats, as we know, come in different shapes and sizes and make up different foraging guilds. And so to look at this relationship a bit further, we looked at how different bat foraging guilds were related to megaherbivore use. And within this model, we found that that significant indirect relationship between megaherbivores and overall bat activity was primarily driven by the positive association with megaherbivore use and the activity of open air foraging bats. And in this study system, those bats are represented by two molluskids. It's the Roberts flat-headed bat on the left-hand side of your screen and the Egyptian free-tailed bat on the right-hand side of your screen. So these open air foraging bats are really looking for clouds of insects in open habitats. They tend to fly quite fast and maybe slightly less maneuverable than the bats that you would associate that feed in vegetation clutter. And so perhaps by having more megaherbivore activity at these sites, we're actually able to see these species in particular have greater access, either for foraging or for accessing the water. Now, during the wet season, I did not find any model that supported significant relationships between megaherbivores and bat communities. This wasn't so surprising to me in the sense that having running rivers and rainfall over the landscape really releases these animals from their need to concentrate around permanent bodies of water. And bats in the system may be tracking highly ephemeral insect resources during the wet season, as pictured here. This Roberts flat-headed bat was feeding over a flowing river the night before. So this is dried mud that you're seeing on his nose that I kindly peeled off before releasing him. Megaherbivores in this system may be searching out different areas where there's greener or new vegetation growth, and perhaps these ephemeral bodies of water that are also come with the rains. So this may cause a disconnect in the spatial distributions of bats and megaherbivores during the wet seasons. One thing worth noting is ideally we would have had data on the insect community during our sampling period. And this was something that I did attempt to do. However, the easiest way to be sampling insects during these nights was using a light trap, which can definitely affect bat activity. And so I was unable to kind of get a clean snapshot of directly what's the herbivore activity, the vegetation productivity, the insect community, and the bat community all in the same day. I was able to do 3 of those, but not add the insect community as well. So ideally, further sampling would go out there and try to perhaps use malaise traps, which require quite a bit more effort in this area, or be sampling on consecutive nights if there is a larger crew, to really look at those direct relationships between bats and insects and vegetation in the system. So in conclusion, what we found is that the conservation of megaherbivores, particularly during the dry season in this landscape, may indirectly promote bat conservation and biodiversity. And given that we know very little about bats in this landscape, but already act to conserve megaherbivores, this was a really great finding. In more mesic environments, however, like savanna ecosystems, it's found that the density of megaherbivores can be high enough that they negatively impact insectivorous bird communities. But perhaps in this desert ecosystem where densities are lower, they're actually able to benefit these less studied insectivorous bat communities. And I'd just like to highlight that the conservation of megaherbivores in these ecosystems does not come without consequence to the people living in these environments. And so we also partnered for my PhD research this with a social study in which we were interviewing local pastoralist communities to understand what are the consequences of living not just with megaherbivores, but also with bats. And we found through these two studies that bats are probably not extremely threatened by people living in these environments. However, there are consequences to living with elephants. They destroy water infrastructure in this area and do threaten human and livestock safety, as well as destroying small crops in this area. So any future work that's really pushing for the conservation of species like elephants in particular should really take into account the interactions that people are having with these species on the landscape. And so I just wanted to highlight that while we're here in a global pandemic, and I'm really grateful that you were all able to join me virtually, bats have really been kind of portrayed in a bad light in recent news. And yeah, there's quite a cool number of interactions that these bats have with other species, both in terms of benefiting people in the landscape with their ability to control nocturnal insect populations and pollinate plants and disperse seeds. But also it's just really interesting when you look at some of these really remote and old environments, as we have here in the Namib, the types of different interactions that occur between species. So with that, I'd just like to thank a number of different people, as well as my funders at the top of your screen there. And I've got my contact information there in case I don't reach your question today or you're watching this video at a later time. Always feel free to reach out with any questions. So yeah, I'm happy to take any questions you have right now. Thank you.
Thanks, Theresa, that was great. Is there anybody who would like to start with a question? Before I go to the chat, maybe if anybody would like to unmute.
Hi, here's Claudia. I could unmute and just read my questions just for the fun of it. That was really a great talk. Many things I had no idea about, so very interesting. What was actually your control group before asking the questions I was writing down? I mean, you had different scenarios, but what is sort of your zero scenario to what you compared it against? Was it dry versus wet season, or maybe I just sort of didn't get that?
No, that's a great question. So at these different water holes, there's really a variety in the amount of megaherbivore use, where some of these river systems have higher densities of elephants and some of them have almost none. It's very transient use in those areas. And so there's a variety of different uses. I would say that all of them have some use during the seasons, but some of them are extremely low. And so the interest here was really comparing not just among those different sites, but also between the different seasons, because as I mentioned, we expected quite a big difference between the wet and the dry seasons there, where we thought there'd be more animals around water holes during these dry seasons and perhaps more intense interactions, and perhaps less so during the wet season.
Right, okay, thanks. And then sort of, there seems to be a beneficial influence of megaherbivores on bat activity, but is there sort of a cutoff density or so, a point where that might turn around?
So we did not detect that in our study here, which was really interesting because that was kind of what I was anticipating, given the findings that you have in these savanna ecosystems where bird communities, I think, really benefit from having some megaherbivore activity in these sites, but then once they get too great, the whole landscape changes in a way that does not benefit birds. And I was wondering if we'd have the same patterns. We did not see that here, and I wonder if it's just the densities are so low that we didn't reach that, or perhaps this is an entirely different system that we're dealing with here.
Thank you. I think I'll hold back because it's not just doesn't make sense. Only one person asking. Thank you very much.
Thank you.
Thanks, Claudia. Is there anybody else who's come up with a question? Maybe while we wait, Theresa, in one of the early slides, you of course made mention of other herbivores also in the system, be it the zebra or the springbok or gemsbok or even livestock. Now, this paper just focused on the megaherbivores, but in the larger project, did you kind of look at those other herbivores too?
In the larger project, I did briefly look at that. There's so much activity when it comes to springbok and gemsbok in this study that I don't think you actually get those same differences that you do in the megaherbivore communities. And even the same with your Hartmann's mountain zebra are quite abundant throughout the study area. And so I wasn't really getting any of those strong changes there. And it's really the megaherbivores, particularly the elephants in the system, that are going to be changing how a mopani tree is shaped in this area. And so that's why I chose to focus on megaherbivores for this study, but I definitely did do a quick check to see, are there other relationships? Particularly, it would have been great if I had more long-term information with where livestock are present, because I think they could also have quite big impacts just in that their density is usually much higher than other species. But unfortunately, I didn't have that available, and it would have been cool to maybe put some like GPS collars out and know exactly where different herds of cattle were, but it just wasn't in this study, maybe sometime in the future.
Yeah, no. Maybe to link on to GPS collars, I know Claudia in the chat had a question about tagging bats. Claudia, would you like to?
Yeah, thanks, Martin. I have not the simplest idea if that is at all possible or practical. When you said that sometimes you record these noises and you don't know, is it one bat or several, or would it make sense to tag individuals and then just have data from them what they do the whole day, apart from hanging in a cave?
That would have been like a big dream of mine in this study. So it would be not just informative for where they're foraging and how far they're traveling each night, but also where are their roosts located, which for the large part, I don't know where most of these bats were occurring. And it is quite possible that bat activity could just be due to the fact that some of these sites were closer to bat roosts. And so that's definitely a follow-up. Unfortunately, these bats, for the most part, were very small, the insectivorous bats. And so we can't really do much in terms of GPS tagging there unless we were able to recapture the same individual and download the data that way. People do use little VHF transmitters on bats as well. But for this study, it was just me and one field technician out in one vehicle. And given that we definitely were having encounters with elephants and lions occasionally at night, it didn't seem safe to try to split up and determine which way these bats were going. But yeah, especially if we could get like something towards the equivalent of like a Modus tower system, where you're able to kind of record some of these bats passively as they pass by. Okay. That would be really interesting. And yeah, if there's any other bat people on this call that want to go and hang out in the desert and help me track bats, I would love that.
Implanting any kind of devices is not possible. They are too small for that, or?
So you could probably use pit tags in them, but then you would need to have pit tag readers up and know kind of where they're visiting. And so that may be useful around these water holes to know if the same individual is visiting the water holes, but without knowing where exactly their roosts are. And actually many of these species are crevice roosting bats, which makes it extremely difficult to do any sampling.
Yeah.
It would be tough, but yes, definitely too small for any of the cool GPS technology that they're using with fruit bats and other parts of Africa.
Thank you.
There's also a question from Sonali Chauhan. Sonali, would you like to unmute? Or do you prefer me reading the question? Okay, maybe prefer. So Sonali says, a beautiful landscape and amazing research. Will you categorize elephants as a flagship species for this landscape?
That's a great question. So in terms of a flagship species, like a keystone species, I would say that it's quite possible, but I don't know if I'm the best person to make that call. There's definitely other people who are studying elephants in the long term in the system and would be able to address more of their long term impacts. What was really interesting to me is my co-author, Joel Berger, he formerly did research in the Namib on rhinos a long time ago and found that they're not really great umbrella species in this area. So by conserving rhinos, we aren't necessarily conserving a whole suite of other species, as you would hope with a large herbivore. And it may just be due to the fact that resources are so scattered in the desert and wildlife have different needs, that conserving one species here isn't going to be the best approach to conserving them all. But I would assume that elephants, there's definitely a strong browse line, and vegetation in river systems with elephants at higher densities is very different than vegetation in other river systems. But this could also just be due to geology and the different soils in the area. So this is something that would be fun to answer, but I'm not the best person to answer that right now.
Sonali also has a second question, and that is, can camera traps be helpful in recording bat abundance?
That's a great question as well. I have tried to work with, I've been working with a PhD student here in the United States on nectar feeding bats, who's trying to look at whether camera traps are useful for monitoring bats over plants that we know they should be visiting. And the fact is that I don't think the technology is perfect for this right now. I think you can occasionally get this, but I think bats move so fast that especially these nocturnal triggers oftentimes will miss the bats passing through the screen. And so it could work, but rather if you're just interested in bat activity and not species specific bat activity, which is all a camera trap would record anyways, the acoustic recorders are definitely the way to go there.
Thank you. Anybody else who would like to unmute and ask a question? I was wondering, when you spoke about the 120,000 odd bat passes over 46 nights, I mean, that sounds like a lot, but how does that compare with, say, New Mexico where you worked, or other arid areas?
So that can be totally variable. And I think it is a lot of times related to not just the distance from a roost site, but also the size of roosts. And so in New Mexico, for instance, is where New Mexico and Arizona in the United States have our largest bat diversity. you can get much larger colony sizes, I would say. And so it really depends on which sites you're sampling. I think in general, targeting bodies of water in desert ecosystems is unarguably like the best way to go if you're interested in getting the highest number of species and the highest number of passes. Bats definitely do cluster at these limiting resources, but this can really vary. Ara Monogem had a really interesting paper out though several years ago that was looking at diversity in Southern Africa of bats, and found that the Namib actually had, I think, a bit more than would have been anticipated otherwise in just an arid ecosystem. So it was a really cool system to work in, and with a couple of endemic species that are not very well studied. But yeah, in terms of number of bat passes, when you're working in systems with like the Brazilian free-tailed bat that we have here in the United States that roost in colonies as large as in the millions, the activity levels really vary depending on the species and the size of the roosts in these areas. And the Namib does not, as far as I know, have these huge roosts that we can find in other arid systems.
Claudia, did you still have a question in the bank there that you?
Yeah, just one. If it's somehow sort of species dependent, or do you have an idea on that? If it makes a difference, if the megaherbivores are elephants or giraffe or rhino or a mixture of them, or because they graze or browse different areas of the trees and stuff.
I think it's mostly driven to elephants being in the system, but given the fact that we had such a small sample size, I didn't want to count all of the changes being due to just elephants in the system. So that's why I ended up pooling the megaherbivore activity altogether. But yeah, I would assume that they're having the largest impact, particularly if it's for these open air foraging bats, which are really dependent on having those open environments, both for access to water to come down and drink, but also for foraging habitats. So yeah, that's what I would guess based on our findings, but without more data, I don't know if I can say that conclusively.
Right, thanks. It might be interesting to see sort of with all this climate change being on the agenda now, how bat density changes, of course, if the environment changes. As well. Yeah,
definitely.
Thank you.
Thank you.
So more waterholes and more biospheres in the Namib will benefit bats more?
It's an interesting question, because actually those artificial water holes, while they harbored more activity, there were like 6 or 7 species that I only detected at natural springs. So just by inserting more water holes in the Namib, we don't necessarily benefit all bats in these systems. So I don't know. It's a good question. And unfortunately, you can't kind of exclude elephants or any species really from those water holes in the Namib. Not only would it not really probably be ethically right, but yeah, good luck trying to keep an elephant away from a water hole in the Namib.
Now that's a valid point. And I was actually wondering, how do you, because you made the comment in the paper where you have those 60% change in the NDVI, and at site-specific level, there was no difference in the megaherbivores visiting the waterholes on a seasonal basis. So it's not about the vegetation, it's about the water that the animals are coming there, is my interpretation of that. And so I was wondering also, how do you tease that apart for the bats? How much of it is the water and how much of it is the megaherbivore? And so I was wondering about potentially, if there are any water holes where you don't have the megaherbivores visiting, but as you say, elephants you can't exclude.
Yeah, And there were, as I said, there were sites where you get really low use by megaherbivores. But these are also sites that may just be very different in the sense that some of them are very far from any vegetation, and trying to have good comparison among sites was tough in this case. I always thought it'd be interesting to try to put out some water and see, do bats come there? But this is also really hard to do in the Namib because of the evapotranspiration rate that occurs there. Like, you'd have to be there adding water every night to have water present. So yeah, I think the best thing to do would just try to be to work with the conservancies and see if they're inserting any new boreholes in the area. If you can kind of do some of the the before after sampling and see how that changes not just the herbivore use of a landscape, but also the bats.
Yeah, cool. So great presentation coming through in the chat. Really amazing research. Is there anybody with a remaining question? Maybe one last question, please. Leabona?
Yeah, this is Leabona, student here at Nelson Mandela University. I just wanted to ask is there any unique niche that the bats fulfill in that system that no other species would be able to fulfill it if they were to be extinct?
Did you catch all of that, Martin? I'm sorry, there was a bit of a...
No, I didn't. Leabona, would you like to try again? Sorry.
I'm asking is there any unique niche that the bats fulfill in that system?
Gotcha. If there's any unique niche that the bats would fill in that system?
Yes.
Okay. So for most of the insectivorous species that we know a lot about, this includes birds, lizards, scorpions, whatnot, most of them are active during the daytime. And so bats are kind of the primary nocturnal predator for insects in this ecosystem. And if we extend that to the fruit bats that are present in the system too, you'll note that they could be doing quite a bit of seed dispersal for some of these fig trees that are really restricted to areas around water holes. So they definitely have unique roles in this landscape. And if we were to remove bats, I'm sure that this would change not just the insect community, but also the vegetation communities that are in these areas. And so actually with a bit of the modeling work, I was interested in whether just the presence of megaherbivores may be attracting bats because they have species like biting flies and whatnot associated with them. But it seemed like the strongest link that I was seeing through the modeling is actually the relationship was more driven by indirectly through their effects on vegetation. So yeah, that's a great question, but they definitely are playing pretty strong roles in this area. And I don't think there's a lot of competitors other than other bats for that niche.
Okay, thank you.
Thanks, Leabona. Thanks, Theresa. I think we need to wrap up. So thank you very much for your presentation. It was really good. And for the time that you spent in preparing, despite everything else that's been going on the last couple of weeks. So thank you very much.
Yeah, thank you, everyone. And I just moved across the country is what Martin's talking about. But I'm glad I could join you today.
And thanks also to all of you for joining us and for the interesting questions that you posed to Theresa. Then just a reminder that next week we have another seminar, and that is at 8 GMT in the morning. And that is by Maria Milicic. And the topic next week is insect threats and conservation through the lens of global experts. And if I'm not mistaken, that was a recent paper in Conservation Letters. So hope to see some of you at least next week, Wednesday at 8 GMT in the morning. Thank you very much.
Thanks, Martin. Thanks, everyone.
Thanks. Bye.